Welcome everyone to Wednesday Night at the Lab. My name is Dylan Brewer and I'm a student worker here at the Biotech Center. On behalf of the Biotechnology Center, UW-Madison Division of Extension, PBS Wisconsin, Wisconsin Alumni Association, and UW-Madison Science Alliance, welcome to Wednesday Night at the Lab. This is the last Wednesday Night at the Lab before long-term hiatus. It is my pleasure of introducing Fernando de la Torre. Tonight he will be sharing about Vaccinium hybrids. Before we get started, we're going to ask him a couple questions. Fernando, where were you born?
I was born in Los Angeles, California, yes.
And where did you attend high school?
Chula Vista High School in South Bay, San Diego.
All right. Where did you go for undergrad?
I went to UCLA.
And what did you study there?
It's a big, long title, but molecular cell developmental biology.
Oh,
we
wow.
call it MCDB, that's what we
There
call
we
it.
go.
Yeah.
And where did you go for any advanced degrees?
Well, I'm currently doing that now, here.
Awesome, good place to be.
Great place to be, yes, absolutely.
All right, ladies and gentlemen, please join me in welcoming Fernando de la Torre.
Good evening, everybody. You know, I was just introduced. My name is Fernando de la Torre. I'm excited to be giving this public lecture to everybody today. If you haven't done so, feel free to take some cranberries and blueberries here. We're going to participate, if you want to participate in a tasting later. I guess I'll start by saying that every time I tell people about my research that I cross blueberries with cranberries, 100% of the time people ask me, what do they look like? So I'm just going to kick us off and show you what they look like. So you see in the middle, actually on the top right-hand corner is a cranberry, if you know, and in Wisconsin we know them very well. And then below that a blueberry, and then in the center there we have a blueberry by cranberry hybrid fruit. And you might appreciate that, you know, they don't really quite look like any of the two parents. They look quite intermediate. And so I'll be talking more about these plants later on in the presentation. But I think it's fun to talk about berries with people. I think most people can use berries to conceptualize something very common that we do in agriculture, which is called hybridization. And hybridization is one of the methods that plant breeders use to systematically improve the edible parts of crop plants for human consumption. And hybridization can lead to the increase of size or flavor of a berry, or improvement of similar characteristics. So like I said, with berries, it's very easy to visualize because I think people intuitively understand that to breed a blueberry with a cranberry is like trying to bring together the best qualities of each into a single fruit. But to accomplish hybridization, we have to mate two distinct plants. And that hasn't always been easy for blueberries and cranberries, and the path forward wasn't really revealing itself until about 5 years ago. Even though blueberries and cranberries are very closely related species, they are sufficiently distinct to deter interspecific hybridization. So interspecific hybridization is a term that we use to describe when two distinct species come together to produce offspring. The blueberry and the cranberry are two distinct species, and the challenge of interspecific hybridization, especially with two fruits as unique as a blueberry and a cranberry, is to find two parents that can come together and produce enough hybrid offspring that then we can select from. So that's the story of what we do at the USDA Cranberry Genetics and Genomics Laboratory here in Madison. We dedicate some of our resources to finding plants within the group called genus Vaccinium that includes cranberries and blueberries to find compatible pairs of plants to produce hybrid offspring. And one of our goals, of course, is to produce hybrid populations and extend them into second and third generation so that we can select the best ones for cultivation. So that plant that you see there is the one that I have here tonight, although I guess 3 weeks later. Yeah, so the objective generally seems pretty straightforward, but it has actually taken decades of systematic testing and cross-pollination to find the compatible pairs of plants. And the blueberry by cranberry hybrid population that I'll be talking about near the end of the presentation is the product of finding two of these compatible parents. So with that said, I want to accomplish three things with this talk. One, I want to introduce the group of plants that contain blueberry and cranberry called genus Vaccinium, and I'll cover a brief history of blueberry and cranberry crop improvement. Then we'll also explore why we would want to breed a blueberry and a cranberry to begin with. Some people might say they're fine as they are. But for that, like I said, I invite you to take a Dixie cup with some berries in it, and then we can go through a tasting exercise. And then we'll also cover the last 30 years of interspecific hybridization attempts. And then lastly, I'll show you some of the measurements that I've gathered on the population of blueberry by cranberry hybrids that I work with. And like I said in the announcement, I will be picking some random people to try a hybrid berry here tonight. And so that'll be at the end of the presentation. So yeah, let's just get started here. As I mentioned previously, blueberry and cranberry are both plants within the group of plants called genus Vaccinium. And the commercial blueberry that you buy at the store goes by the common name highbush blueberry, and it has a Linnaean name of Vaccinium corymbosum. And the commercial cranberry goes by the common name American cranberry, and its Linnaean name is Vaccinium macrocarpon. This genus is very special to us because it's a group of plants native to North America with distribution along the East Coast and in the northern regions of the continent. This is very different, for example, than tomatoes or apples, which originate elsewhere in the world. For example, tomatoes are originally from the Andean region of South America. And as much as we associate tomato sauce with Italy, tomatoes were not part of Italian cuisine until the Columbian exchange in the 16th century. Similarly, apples were originally from Central Asia, east of the Caspian Sea. And in fact, there was once a medieval settlement near the capital of Kazakhstan called Almatau, which means apple mountain. But cranberry and blueberry have their centers of origin in the northeast of the USA. This allows us to directly observe these plants in their natural population, in their natural habitats, and select from the original gene pools with minimal geographical and political constraints. So to talk about Vaccinium and the earliest indications that we could potentially breed between them, we can start by talking about Canadian botanist Sam Peter van der Kloet. He and his family immigrated to Canada from the Netherlands in 1947, and he would eventually become professor at Acadia University in Nova Scotia and one of the top authorities in blueberry classification. And if one of the really famous books that he wrote called The Genus Vaccinium in North America, in reading that book, you start to realize that he was very frustrated with blueberry classification at the time that he finished his graduate degree in 1972. In the preface of this book, The Genus Vaccinium in North America, he noticed that twigs from single colonies of lowbush blueberries, both in New York and Ontario, when fully exposed to sunlight, were sometimes referable as Vaccinium brittani, when partially shaded as Vaccinium angustifolium, and when completely in the shade as Vaccinium lamarcki. This is patent nonsense, he said. Any existing classification that gives such ambiguous answers brings taxonomy into disrepute. So what he's trying to say with this is that he believed that the system at the time was a source of many misidentifications in the genus. And so for this reason, he went out to document many blueberry and cranberry Vaccinium species across the continent and extensively surveying natural populations. He collected continuous characters. So what do we mean by continuous characters? They're traits such as corolla width and length, which captures the shape of the flower, the crown, and the petals. Anther lengths, which the anther is the flower organ that produces the pollen, and as well as the diameter of the pollen itself, which, if you can believe, varies across the different species. And so he did this for 184 Vaccinium populations in North America between 1969 and 1981, and he also collected soil samples and took notes of the habitats. So, I mean, with that much experience, it makes sense why he became an authority in the field. And so based on the distribution of the vaccinium populations and the soil that he was gathering, he proposed an origin story for the genus Vaccinium, which was in some agreement with his predecessor, a really important botanist called Wendell Holmes Camp. And so the hypothesis involves the last glaciation period of the Quaternary, which is also called the Pleistocene glaciation. And many of you are familiar with this concept because of of how glaciation forged the terrain here in Wisconsin. So from studying ice cores, geologists have described this period of Earth's history as having glacial and interglacial periods, as you can see up there, where the ice would nucleate or start from the American Cordillera and then proceed towards the 38th parallel. And that's a line of longitude. And so during interglacial periods, the ice would go back towards the mountain range, and then during glacial periods, it would advance again. And this happened several times, and each time the ice sheet advanced, it displaced many plant populations east and south, and also flattened much of the land, which is what you see across Wisconsin. And so the interglacial periods might have been responsible for promoting the evolution of proto-vaccinium populations. It's a complicated story to tell, but I just wanted to give you a sense for the natural selection that took place by the time that van der Kloet and Camp and other botanists surveyed North America for these populations. I should also mention that vaccinium populations do exist as far south as the Andes in South America and on islands across the world. And this can be explained by migrations that occurred much earlier in Earth's history, where there were land masses that are non-existent today. But that's beyond the scope of this talk this evening. So ultimately, what I want to get at, what is relevant to us tonight, is that van der Kloet and Camp, they found naturally occurring hybrid populations in their surveys. And so here I'm showing a couple of independent examples of hybrid zones described by these botanists along, Florida and New York. And the hybrid zone is a region where two distinct plant populations can cross-pollinate, generate hybrid seed, and have that population take over a region of the geography. And so the two plant populations need to be something we call sympatric, so they have to flower at the same time and they have to have a pollinator that they share for the cross-pollination to occur. And so vaccinium hybrids are still found naturally today, and there's a recent example in the San Bruno Mountains in California there up top. So given what van der Kloet, Camp, and others did, we had a better understanding of how we organize vaccinium plants today. We have the main genus Vaccinium, and then it's further subdivided into subgenera called sections. And when van der Kloet and Camp were doing these surveys, they only had morphology to work with. And now, all of us know we have DNA technology to help clarify the relationships between different plants. And so the most comprehensive work that was done on this front was by a professor called Kathleen Cron. And right before I get to that, these are the kind of maps that van der Kloet and Camp could put together, given the distribution, where they found the plants, and then the morphology that they shared between them. But again, this is not complete, given that we have DNA information today. And so Kathleen did a lot of this work as a professor of biology and molecular systematics at Wake Forest University. And she spent her whole career studying a larger family of plants called the Ericaceae, which contain the lineages that gave rise to Vaccinium. And so you can see the plants that she surveyed shaded on the world map, and then these more sort of refined relationships that she was able to decipher with DNA. So returning to that diagram then, because with van der Kloet, Camp, and Cron, now we have a better idea of how the genus is subdivided. These hybrid zones that Camp and van der Kloet described were between members of the section Cyanococcus. And so cyano means blue, and coccus means sphere. So you can already infer what that means. It's all the blueberries that you see naturally and at the store. And so you might be, at the time, this was the best example that we had of being able to cross between species in the same section. And it's the reason why we have blueberry in store shelves today. And so I'm going to go over a little bit of the improvements that were done to blueberry, quick history. And we have to start in 1908 with Frederick Covell and Elizabeth White, who was the daughter of a cranberry grower in New Lisbon, New Jersey. And Frederick Covell was a USDA botanist. And up until 1908, you could only harvest blueberries from the wild because farmers didn't know how to culture them in garden soil. And so in his book from 1910 called Experiments in Blueberry Culture, Covell describes the experiments that he performed to ultimately discover that blueberry plants need acidic peat that is not overwatered to grow successfully. And so peat is a compost of leaves and wood and moss that can be found in bogs and surfaces of pine woods. And so Elizabeth White read the book and was very excited about the opportunity of growing blueberries commercially, because they weren't doing that at the time. And so she offered Covell the use of her land in New Jersey as well that was ideally suited for blueberry growing, because that's where blueberries came from, and her labor force to begin the first breeding efforts to increase the berry size. To provide a breeding example, Dr. Covell brought a wild selection that he had from a neighbor called Brooks, and he crossed it with a wild selection that White made near her farm called Soy. And then they would cross them and then evaluate the size of the berries, take really careful notes. And Elizabeth White continued breeding way after the partnership between Covell ended. And so I just want to clarify a point here with wild selection, what I mean by a wild selection. And so I guess yes, here we go. Yeah. So wild selection is simply a plant chosen from a naturally occurring population. So here's a little short video of wild cranberries growing near a lake at the Kemp Natural Resources Station near Minocqua, Wisconsin. And if you wanted to make a wild selection of these cranberry plants, you would go grab some vines and propagate them based on something that you observed, like better berries or vigorous growth. So that being said, cranberry culture was mainly based in wild selections until the initial cranberry improvement cycles were undertaken as a response to false blossom in 1929. That's very different than blueberry, because in blueberry they wanted to increase the size, but in cranberry there was a disease that took over. And when false blossom emerged, it threatened the cranberry industry because it didn't have a cure. So false blossom is caused by a bacteria that is carried by an insect called the leafhopper, in particular the blunt-nosed leafhopper, and it transmits a bacteria that prevents the cranberry flower from assuming its down formation. You can see how it kind of has the cranberry, a healthy cranberry inflorescence will have like a crane formation, and when it's diseased, it won't be able to assume that formation, and therefore the fruit can't develop properly. And so, so the two researchers that wanted to find a solution to this problem were R.B. Wilcox and USDA entomologist Charles S. Beckwith, because at the time, the industry by 1929 had already advanced beyond Massachusetts and New Jersey into Wisconsin. And so if you have a disease that's out there that has no cure, it can potentially decimate the local economies and people's livelihoods. So what they were able to figure out, Wilcox and Beckwith, was that some of the wild selections that were actively grown in Massachusetts, called McFarland and Early Black, were the most resistant to this disease. And in a future improvement cycle between 1934 and 1950, the cultivar Stevens was shown to be the most resistant, which came from a cross between two wild selections, one from Massachusetts and one from Wisconsin. So I want to note that even though Coville, White, Wilcox, and Beckwith were very successful in their improvement efforts, those improvements were largely regionalized to a certain extent, because even though blueberries could be cultured, they still required cold temperatures to break out of dormancy. Because now we know that as fall approaches and day lengths shorten, less light is available for plant growth. And so for this reason, the plants enter dormancy to slow their growth, strengthen their roots, and conserve energy. So the prolonged exposure to cold temperature is important for breaking out of dormancy, because it begins the process of releasing sugar stored in starch in the plant cells. And then as winter ends, the day lengths increase, and then the plants can use this starch to promote their growth in spring. So at the time, now we know this, but at the time, during Coville's improvement cycle for blueberry, people thought that if you just put a plant in growth in a warm temperature, that it would actively grow. And so Coville showed that chilling hours were important for blueberries to exit the dormancy. So then how did we go to bring it all back to interspecific hybridization? How did we go from a regional crop like a blueberry that only grew in the Northeast to now what is the global production of blueberry? And that actually was through the interspecific hybridization of highbush blueberry with the evergreen blueberry. So these are two plants in the same section, and the evergreen blueberry up top had been selected naturally by the temperatures in Florida. This is a Florida native. And so if you could cross the blueberry that has a higher requirement for chilling hours with a blueberry that has a lower requirement for chilling hours, then you could have progeny that you can select from that have lower chilling requirements. And so that's essentially the answer. So that interspecific hybridization between two different species in the same section led to what is now global production of blueberry. Right, so breeders seeing that interspecific hybridization was possible within a section. So just to clarify, we have different species in a section, and then they're able to cross amongst each other, right? But the genus has so many sections, so much variation. So plant breeders really wanted to know if they could, cross between sections to be able to harness the speciation across the entire genus. And so why would they want to do this? Like I posed a question earlier, if blueberry is probably fine, a cranberry is probably fine. But if you look across the whole genus, you can potentially unlock a lot of variation and customize different berries for different climates, which would be really helpful for, for nutrition and berry enthusiasts, if there are any out there. Right. So now I've been talking for a few minutes, and I wanted us to explore the tasting. I'm not sure if any of you have berries or if you've eaten them yet. So I'm gonna take a couple here. And I want us to try a cranberry just first on its own. It doesn't get any easier. And can I get some responses, some one-word responses about what people are? Yeah, very tart.
Are these all the same?
Yes, they're all the same. Yes. Anything else?
A little bitter sometimes.
Yeah, crunchy. That's a good one. Yeah. Right. So I think that's. So you can kind of see why we would want to make improvements by hybridizing with other species. So let's just try a blueberry to, clean the palate. Exactly. And so what are you tasting here?
Sweet.
Yeah.
Can you repeat the responses?
Yeah. So sweet. Somebody said sweet.
Mushy.
Mushy. True. True. What was that?
More chewable.
Yeah, more chewable. Absolutely.
Juicy.
Juicy. Yeah.
Yummy.
Yummy. Yeah. That's exactly the point. So now let's just eat them both together and see what maybe potentially, how, if you took genes from both of them, could you improve the experience here? Yeah. I didn't grimace that time. Okay, well, thank you for participating. I'm glad, I'm curious that nobody brought up the skin of both fruits. The formal term is a cuticle, and that's the barrier between the interior flesh and the outside world. And there's this waxy layer called, there's a layer of wax and some biological polyester called cutin. And so you might have noticed that after buying blueberries at the store and leaving them in the refrigerator for a little bit, they start to shrivel. And so you might conceive that a blueberry could be improved by taking the cuticle of a cranberry, which doesn't really lose water. You can keep them for weeks, and they'll still be as firm and tart and sour. But a blueberry doesn't do that, right? And so that could be an improvement made to blueberry, right? You improve the cuticle and you extend shelf life. And then we don't have to go very far in terms of the cranberry. It's tart, it's very sour, and the sugar in a blueberry could really improve the taste for the fresh market, because most of us are familiar with cranberry through our cultural experience of Thanksgiving and a sauce, but that has a lot of sugar. So for the fresh market, there can be an improvement. Right. So let's see. Yeah, so these are the combinations that I was referring to when I was talking about unlocking the potential across the genus, right, between all the sections. If we could figure out a way to cross between them, then we can customize berries for different regions of the world, different palates, etc. Great. So I'm going to go through a little bit of the history of the scientists and the breeders that have tried to accomplish this goal over the last 30 years. So Sylvia Brooks and Paul Irene in 1996 had similar ideas here. So they wanted to take the Vaccinium arboreum, which is called a sparkleberry, and they wanted to cross it with highbush blueberry, because the sparkleberry has a larger growing region than Vaccinium corymbosum, the blueberry. So you could potentially, expand growth and commerce to different areas. And so they did some interesting findings here, because they realized that they couldn't take a blueberry and a sparkleberry and cross them directly together. They had to go through an intermediary called Vaccinium darrowi. And so this was called the evergreen blueberry. This is the one that was used to lower the chilling requirements for Vaccinium corymbosum. So this plant is very interesting. And so they took an intermediate hybrid between Vaccinium darrowi and arboreum, and then they were able to cross to highbush blueberry. So what was interesting about that is that they figured out two things. They figured out that they could cross between sections, in this case, section Cyanococcus, which has all the blue spheres, all the blue berries, and section Batodendron, which is where the sparkle berry is organized. And secondly, that maybe you do need to have one hybridization before your target to have genetic exchange that you want. Then in 1997, Zeldin and McCown here at UW, they used a very interesting plant called Vaccinium reticulatum, or the Ohelo eyeberry from Hawaii. It grows on volcanic soils on the islands, and they figured out some interesting things here because they were able to cross directly between Ohaloi and cranberry, and then Ohaloi and lingonberry. Lingonberry is very famous in Swedish cuisine as in jams and all that. So they were able to cross when Vaccinium reticulatum was used as a mother. So they could only really put pollen from a cranberry or pollen from a lingonberry onto an Ohaloi flower, but not the other way around. So this shows you that there's some unidirectionality in some of the crosses. And so that in itself is interesting. But now, before I continue on with the history, I kind of want to go through some of the mechanics of how you would even hybridize. So I'm talking about crossing these two plants together, right, to potentiate all the variation across the genus. But how would you do that? So here I'm showing that all flowers have a stigma and a style where they receive pollen from a different, from either the same species or another species. And so when flowers are ready to accept pollen, they usually have this little exudate that will hydrate the pollen and begin the process of fertilization. So what do I mean by hydrating pollen? So here on the left, I have dry pollen from Vaccinium macrocarpon, or the American cranberry, the variety Stevens, and then when it's exposed to this exudate at the tip of the stigma of a flower, it will then expand, and this begins the process of the pollen sending down their genes. And so here I'm showing a pollen tube that emerges from the aperture of a pollen grain of Vaccinium crassifolium, and at the tip of this pollen tube, you can see on the right, in the little red pollen tube at the tip are the nuclei that belong to that are the genetic material of the male in the cross. And so this is really genetic transport. And so then as the pollen tube, as the pollen is hydrated at the tip of the stigma and pollen tube elongation commences, it goes down the style of the flower towards fertilization. So here you can actually see I cut a little window in a style and stained it so you can actually see the pollen tubes like up top, but actually in the flower organ. And if that wasn't clear, the style is that little organ that's protruding, for example, from this flower of Vaccinium macrocarpon. And then the pollen tube and the genetic information is traveling down towards the ovules, which eventually become the seeds. And so that's what I mean by fertilization. So that's what you would do in an interspecific cross, is you would take the pollen from one plant, and then you would take the flower of the other, and then put the pollen on the flower, and then hope that the pollen elongates, goes down to the ovules, and eventually gives you a seed. And that seed is going to be the potentially hybrid plant that you would grow up, like you see here. So I'm going to kind of go through this a little more briefly here. There were some really important advances here that kind of still suggested the same thing. For example, here Nikolai Vorsa and Jessica Ciccolesi-Johnson, they figured out that, again, you do need an intermediate. So in order for you to cross Darrowi with the evergreen blueberry with macrocarpon, you need an intermediary hybrid with oxycoccus, which is the small cranberry. And then Mark Ehlenfeld and Jim Polashok, they were able to figure out that somehow if you were to find Vaccinium species that are very distant on remote islands, like for example, like the Ohalo eyeberry, in this case you have Vaccinium padifolium, which is it's on an island off the coast of Portugal, and they can mediate hybridization as well. But I do want to bring up one person here is Paul Irene. So you can see here that from 1996 with Sylvia Brooks, all the way until 2018, he's been trying different hybridizations. And so it takes an entire career of testing different combinations to get the populations. And in this case, Paul Irene he was doing crosses in a very classical way. So here he was taking high bush blueberry, which is again the one that we eat, and he was crossing it to deerberry, which is the picture I have there with Vaccinium staminium, and he was generating a first generation hybrid that he would then use to the pollen from to back cross to the high bush blueberry. And this is how you get a little bit of genetic exchange, but you mainly maintain the identity of the plant that you want, which is a high bush blueberry here. And so it takes a long time, right? And so now, in the present, this is the real development now that is the core of my graduate studies here. And it's here in 2017, Mark Ehlenfeld and Jim Ballington would find a plant that, for reasons that are still unclear, would be able to cross to both high bush blueberry, lingonberry, and cranberry, which are all the commercial species. So this is exactly what we'd been looking for, a plant that could be a mediator for genetic exchange across the entire genus. And so I just wanted to point out this plant here. It's called the Andean blueberry, or Vaccinium meridionale. And so you find it in the mountains of Antioquia in Colombia, and people call it there in Spanish, they call it agraz or mortiño. And it's found in the Colombian Cordillera, or basically the Colombian part of the Andes. And you can see there that what the plant looks like. And so for a long time, this plant was basically harvested from the wild. And so I wanted to put a human element to this. A lot of the local people there didn't really, they would just use it to eat and not really realize that it could actually have this implication in science. And so it's very interesting. The only thing I wanted to point out here is that now they're going through the process of commercialization in the same way that Elizabeth White and James and Frederick Coville did in the 1910s, where they were creating markets for different berries. And that's how, now with the right markets, they're able to then fund their breeding programs and actually make it something that's cultivated on a farm, rather than just it being wild and for personal harvesting. And so Mark Ehlenfeld and Jim Ballington, they were able, the first thing that they figured out is that the Andean blueberry can generate many hybrids. So generally speaking, in previous attempts throughout the years there, you can see that the frequency of getting a hybrid by an interspecific cross is very low. I mean, 4 hybrids in 3,300 crosses. I mean, those are pretty slim odds there. And so with Vaccinium meridionale, they were able to get way better odds, 190 out of 1,900. And so why is this important, right? And I'll get to that in a little bit, but basically the odds, just to drive this point home, the odds now between a cross or the frequency of generating hybrids between Vaccinium arenale and macrocarpon now is 500 F1 hybrids per 30 pollinations. That's something that you can do in 5 minutes. And so why would we want so many hybrids? Here the idea is that for a hybrid to be successful, you need a large starting population. Why? Because you need all the genetic combinations. You want to generate enough genetic combinations that can display the full array of all the characteristics that you want to see, so then you can select the ones that are going to be the best. And not only that, but you also need the plant to be self-fertile. So you need it to produce fruit, and you need the fruit to have seed, so that you can advance the hybrid populations into a second and a third generation. Right, and so now this part of the presentation, I'm going to start talking about this F1 hybrid that I'm studying here, thanks to plants that Mark Ehlenfeld has sent over from New Jersey. And basically, it's Vaccinium arenale by Vaccinium macrocarpon as an F1 hybrid. And something you can see that's quite odd is that the berries don't quite look like the mom or the dad. They don't look like the cranberry or the blueberry, right? It's kind of in the middle. Yes, so now I'm going to go into the metrics, like I said. So what am I seeing? What are we seeing here with all the hybrids that are being generated between this cross? And so they all come in different sizes. And after about 2-3 years of growth, the sizes have been consistent. So there are some seeds that are going to be generated that are never going to produce a big plant. And then there's some seed that is going to produce a big plant. And that's exactly what I meant by large population, because then we get all the genetic combinations for selection. Furthermore, not all the F1 hybrids are flowering. So that is one of the things, those are one of the requirements for being able to select a good hybrid. You need the hybrid to have fruit so that you can take the seed and continue advancing the hybrid population. And then lastly, not all the F1 hybrids produce fruit. But the good thing is that because we started with a large population, we have about, right now we're hosting 200 or so plants here of these F1s from the original 500 that were generated from the 30 pollinations. And I think it's like 40% of them are flowering. But had the cross only generated a small amount of hybrids, you would have less plants that are flowering. So the point is that this is exactly why you want large populations. And then the curious thing here, and the reason why we're going to continue advancing the populations is because not all the F1 hybrids are producing the same fruit. So some of the plants will only produce cranberry-like fruit, and then some of them only the blueberry-like fruit. And then some plants produce a mixture, and I'm not exactly sure why that is. But what you can appreciate here is that the internal flesh is very different. So in the ones that look more like blueberry, you have a red sort of flesh with potentially a softer cuticle, a softer sort of waxy or less waxy skin, versus the ones that are red that have that are more cranberry-like. But then I'm comparing it next to a cranberry fruit, and I want to make a point here because you notice that a cranberry fruit, generally speaking, has holes in it. I don't know if you got a chance when you were grimacing after eating one of these, but yeah, if you, I'm not gonna. Yeah, you can see that it has holes in it, right? And so as we continue selecting, we're gonna have to take this into account because it actually plays a really important role. We don't want to disturb agronomic practices, harvesting practices, and so many of you are familiar with this image here. As I mentioned before, a lot of cranberry beds, they start off as like single plants on the bottom of a pool-like excavation that has sand on it, and after the flowers bloom and the fruit matures, then they flood the bed, and then the berries will then float, right? But the holes do contribute to flotation, but also the fruit density. It's the same reason why apples float, but they don't have holes. And so in our selection screening, we're noticing here, I'm playing a couple of videos of just dropping the berries. So that's one of the purple berries that the hybrid produces, and the flotation is a little less fast coming back up. But then some of the red berries, they float just as well, even if they don't have holes. So this is one of the things where we don't want to disturb the industry, and so we have to consider that we want to improve flavor, but we also don't want to affect the density of the berry. Something else we do is we test the different levels of firmness. And so here we have this machine. I tried to be as still as I could, but basically it's a machine that will press the berry, and it will generate these curves that will give you the maximum compression force. And so basically that kind of tells you how well does the berry compress, right? And so if it's softer like a blueberry, you know, the curve is going to be lower, as you can see down. All the blueberry curves are really low, right? And cranberry, as expected, is a very firm, very crunchy berry, right? But what's great is that the F1 hybrids are producing variation that we can select, because some of them are softer, some of them are firmer, and so that's what we want, and that's why we're so interested in this cross. And then lastly, the F1 hybrids show vigor for seed potential. So remember what we had talked about was that you need a large starting population, and you need the flowering plants to produce seed. So here, what I did is I dissected out the ovules from different flowers. And so I had mentioned that the ovules are where the pollen tube will finally do the fertilization, and then those ovules will become seed. So if you open up the flower and count how many ovules, you can get an idea of potentially how many seed it's going to produce. And so what you can see here, I compared the two parents versus the F1 hybrids, and you can see something that we call hybrid vigor. And so that's when there's enough divergent genetics that the F1 hybrid will be greater than the sum of its parents. That's sort of like the easiest way to understand it. And lastly, and this is the part where I want to go back to the cuticle example that I had talked about. So I wanted to test if my idea for improving the shelf stability of a blueberry could be possible, right? So with the F1 hybrid, so we know, like I had mentioned, that a blueberry, here I left some blueberries out at room temperature, and you can see that in 6 days they're kind of shriveled, but a cranberry keeps very well, right? And so we have variation in the F1 that we can select, and so I wanted to know if exposure to just room temperature and having it dehydrate, if the cuticle, which I can see here, the blueberry-like one has less of a waxier cuticle than the cranberry one, if that has an effect on the loss of water, or basically dehydration, right? So I was able, at the time, I didn't have the F1 hybrid berries, but I did have this plant here, and so I'm going to introduce, I wish I had a name, but I'm going to introduce this plant here. But it's basically, it's a very similar plant to the original Andean blueberry by cranberry, only this one's a little further along. So the ones that I work with are the F1s that were generated by Mark Ehlenfeld in New Jersey, and then this plant here is the second generation hybrid. So it's a little further advanced. It's been selected. And yeah, so the seed from the hybrid fruit was germinated, and it gave us this plant. And so what you can see here is I took some cranberries, I took two types of blueberries, and I took the hybrid berry, and I just left them out. And you can see here that cranberry hardly loses any water, I mean, at room temperature. And the blueberries just plummet, right, because they shrivel. But the F2 hybrid, on average, doesn't lose water as quickly. And so, you can see that physically in the berry, right? And so I did a very, there's some indication that this is also an effect that is seen in the in the hybrid fruit itself, in the F1 that I'm working with. I didn't have a large sample size, but basically I took some of the red berries and I compared them to some of the purple berries, right, versus cranberry. This isn't a great test because I didn't have a lot of fruit at the time, but you can see here is that the rate of dehydration is higher for a blueberry, for the blueberry-like F1 hybrid than the cranberry-like F1 hybrid. So I felt like that was, I'll be running more tests, but essentially there's promise here to kind of improve the shelf stability of a berry that's a little sweeter. We are working with a USDA food scientist right now to actually describe quantitatively the flavor of the berry. But there's a lot more to do, but I think this is kind of a proof of concept that you can increase the shelf stability by using more of a red phenotype, selecting for a blueberry or a berry that's sweeter but that has more cuticle to maintain its shelf life. So yeah, that's it. And like as promised, I just wanted to have one person randomly come up and then pick a berry, and then now you've tried a blueberry, now you've tried a cranberry, and this is very similar. The lingonberry is very similar to the cranberry. It's just a little smaller. The flavor is a little bit more complex, but this is kind of what you could expect from an improved variety. So I wanted to have somebody randomly come up, any volunteers, just to pick one out and then tell me. Right here, both of you. Yes.
Do you have any tips for picking a right berry here?
No, let's see. Let's pick them on this side though, so that people can see.
Make the faces, Trevor.
Yes, and then just look for something that you think might taste good. The redder, the better. That's probably as much as I can say. So you've tried the cranberry, you tried the blueberry, right? Okay. Okay. So we'll have you go first, and then I want you to describe the taste to the audience here.
Oh, that's good.
Yeah?
It's like similar to the cranberry, kind of in like the firmness of it. Like it has like that kind of waxy skin, and a little bit tougher, but it's not as tart. Like it's sweeter.
Great. Okay, we'll have our second participant here.
It's definitely a lot sweeter than just the plain cranberry. You can definitely taste like the sour aspect that comes through, but it's countered really well by the sweetness. And then, as you said, the cuticle is definitely a little tougher, but it's nowhere near as tough as the cuticle of the cranberry.
There you go. Well, you heard it here first, I guess. All right, thank you. All right, you can have one more and go ahead.
They're good enough, they want it.
Yeah. So, so that's essentially it. I just want to, before I wrap this up, I just want to, thank some important people. These are all my advisors here. Anna Edlin, who has been really helpful in showing me all the techniques for imaging the pollen that you saw, so that I could communicate effectively how the fertilization happens. And of course, Mark Ehlenfeld, who's a research geneticist, a blueberry breeder in New Jersey, in Chatsworth, New Jersey, and of course my advisor here on campus, Juan Zalapa, who is a research geneticist as well, a cranberry breeder with USDA ARS as well. And you can see that this project is complex, so it's unique. So it needs unique fields to be explained and to be able to, hopefully someday potentially be able to cross across all the sections and get, interesting tasting berries so that people can enjoy them. And then lastly, I have a small team here. One of my undergrad mentees, Jordan Cargill. Eric Wiesman is he's a research technician as well with USDA ARS, but Secretly, he's our lab manager, so he really helps with logistics, and we're really grateful. And then our technician, Yvonne Hernandez, who works with us as well. And so that'll be it. I'll take any questions now.